It gives me great pleasure to announce Dr. Jason Gill. The C staff and I met Jason when we went down to Texas A&M to bond with Elizabeth Summers, which you guys listened to yesterday about troubleshooting the DNA prep. And Jason was a pleasant surprise during that time. Jason is a postdoc at Texas A&M. He has been doing phage biology for over 10 years now. He's a real expert, and he's going to talk to us about applied phage biology. Okay, welcome, Jason.
Thanks. Can everybody hear me okay? Okay, so I'd like to try to keep the talk as interactive. I think people are pretty comfortable with each other now. So if anybody has any questions during the talk, you just raise your hand, and then Matt will come around with the mic, like Donahue, and because this is being recorded, so you have to try to capture the questions in the mic as much as possible. So I want to talk about applications of phage biology. Now, as Graham mentioned, a quote from Ra Young, the lab I work in at A&M, that what we know about the genetic level of phage biology is an inch wide to a mile deep. So when you go to the applied side of phage biology, what we know is more like a mile wide and an inch deep. There's a lot of people that tend to become smaller projects, and it's in actually much more complex systems. So I'm going to be going through a number of different facets in which phage biology can be applied. And so I want people to ask questions. If I may not address things, you have particular questions you want to answer that I'm not addressing. So don't be afraid to interrupt. Okay. So the first real application in phage biology was in the 40s in the development of molecular biology. So I'm not going to cover this too much. This is older work, and this is all stuff that students will tend to get in their molecular biology or genetics courses. But phages were instrumental in making most of the early genetic discoveries, like the nature of genetic material. So the work by Lurie and Delbrook, their paper was the original fluctuation test, used phage T2 as their selection agent in order to determine the heritability of genetic mutations in bacteria. The classic Hershey-Chase experiment where phage particles were differentially labeled. The DNA was labeled with B32 and virus was labeled with S35, and cells were infected, and then the attached wire on the outside was sheared off in the blender. And that showed that only the P32, the DNA that was labeled, stayed with the cell. So that showed that DNA was the hereditary material. And of course, Benzer did his work with the R2 mutants, and that showed that you actually have genetic recombination in a gene. And this was accomplished by doing crosses with R2 with phage T4 R mutants. Which give a larger plaque phenotype, which are not able to plate on a certain strain, which is how they could select for recombinants, which had reverted back to wild type. So, does anybody any more refresher on this, or I'm just going to keep going? This is the students will probably actually get this in other classes. At least I hope they do anyway. So I want to talk about a lot about the kind of my field of research, and something which has really become of great interest in the last 10 to 15 years, and that's the use of phages as antimicrobials. So there are pretty much three ways that you can use phages to kill bacteria. And so there's the classic phage therapy, which is you have phage, which attaches to a cell, ejects its genetic material in the cell, directs the production of new virions, and the cell is then lysed, releasing the virions which then go on to kill other cells. And this is kind of the classic form. This was originally actually used in the 20s to treat bacterial infections, and continued on in the former Soviet Union since then, but it kind of died out in the West from the 40s, and it has been a kind of resurgence of interest starting in the mid-80s, and that interest has been going up ever since. A much more recent development is what they call phage methylation, which is essentially a defective bacteriophage. You have a phage which is able to bind to, infect a cell, inject with genetic material, but is unable to lyse this out. And there's a couple ways you can accomplish this, but so what this gives you is a much more defined dose. The phage will not be replicating on the pathogen in the patient, but it will kill the bacteria. And then the third, which is also more recent, is the use of isolated phage lyses, which is the phage endolysin isolated, and these can be used as antimicrobials directly, particularly in gram-positive. So the classic phage therapy is the use of viable phages, preferably virulent phages, as tempered phages tend to form lysogens, and those are not what you want for extended applications because the lysogenic bacteria will be resistant to further phage infection. The phages are lyse their host, and they are very specific, of course, for their host, which has advantages and disadvantages. It has advantages that won't disrupt any of the beneficial flora in the host. And another advantage is phages are relatively cheap to develop. So phages were discovered independently by Twort and Harrell. Twort discovered it first, and I wrote one paper on it as a neat thing, and then didn't really work on it much more, but Harrell immediately became obsessed about it, and continued work, and he almost immediately saw potential for phages therapeutics, because he actually had originally isolated phages from dysentery patients. So this started a field of research which continued pretty much from their initial isolation in 1917, 1918 up until the 40s, and pretty much ended with the development of chemical antibiotics. So here's Phage Biology. Taken probably sometime in the 30s. So Harrell found phage in the stool of late-stage dysentery patients. So, and he had noticed that you seem to see more phage in patients that were going into remission than patients that were early in infection. He hypothesized this actually has something to do with the remission of the infection. So, well, he didn't try directly on humans right away. He did try some poultry experiments where he had a phage-treated, and actually a pretty well-designed experiment for something that was done in, I believe, 1922. Had a phage-treated and untreated control group of chickens. They were exposed to infected birds to inoculate them with a pathogen, and he did note a decreased incidence of disease in the phage-treated poultry. So the early literature on phage therapy is pretty spotty. Now there are a lot of papers done, however the quality of the work is highly variable. There was really very poor understanding of the nature of the phage. There's actually a lot of debate, even up until, well, the first electron micrographs were taken in 1940. So up until that point, a lot of people had, there was a school of thought which believed that they were actually enzymes or some kind of like dormant lytic principle in the bacteria which were somehow triggered by exposure to other lytic principles. And so that's this kind of level of understanding people had about phages in this period. And so the research continued through the 20s, 30s, and into the early 40s. But there was a couple of reports, in particular this one here from the American Medical Association by Eaton and Bain Jones, which came out in 1934, which gave not very favorable overall review of phages and antimicrobials. So, but also this is, you have to keep in mind that people didn't really know what phages were. And actually, one point that the report makes is that, well, we actually think that phages are not even alive, and that they're not actually viruses at all, so this whole thing is hokum. And of course, they were completely wrong, but that still held a lot of sway at the time. And then there's another report which came out in 41 from Kruger and Scribner, which also reviewed the literature. And it gave a better overall assessment, but still, there was still a lot of problems with repeatability. It was not very predictable about when it would work and when it wouldn't. Although it did tend to work better in, say, gastrointestinal and surface wound infections than if, say, somebody had a very deep abscess. Lung infections tended not to cure very well. So the phage